now. So our next speaker is Dr. KMV Narayana Rao, who is the Associate Vice President for Head of Analytical Aragen Life Sciences Private Limited. Since June 2015, Dr. Rao is associated with Aragen Life Sciences, supporting both contract manufacturing services and chemical development services business units. Dr. Rao has expertise in analytical research and development in APIs and NCEs. Key contributor to primary registration documents CMC, IND, ANDA, CEP, DMF. His experience in customer agency inspections and audits. He has the ability to drive projects to success under pressure, responsible for the proactive identification and resolution of analytical procedures, problems, and issues. He has hands-on experience with state-of-the-art analytical instruments like HPLC systems, LCMS, GCMS, ICP-MS, PXRD, ion chromatography, etc. He has provided training to team members in all procedures, instrumentation and techniques, including but not limited to HPLC, GC, UV, VIS, IR, raw material, finished product and stability testing. He has designed and operated well-equipped analytical R&D labs and successfully got the approvals for the regulatory authorities across globes like US FDA, MHRA, PMDA, etc. He has published more than 25 papers on genotoxic impurity quantification, chromatographic methods using hyphenated techniques, residual solvents analysis, high sensitive analytical methods with sub ppm levels in peer reviewed journals. He is also a reviewer in the editorial board of international journals. He has previously worked in Sai Life Sciences, Zydus Cadila Healthcare, Dr. Reddy's Laboratories, Hetero Drugs, Newland Laboratories, and Vimla Labs. We welcome you, Dr. Rao, and over to you, sir.
Thank you, Shivani, and welcome and good morning all. So I would like to take you through some of the technical aspects and requirements on the pharmaceutical grade solvents, and I would like to share my slide deck.
Yes, sir. Yes, sir.
I hope it is visible.
Yes, sir, it is visible.
So use of high purity solvents to manufacture bulk drugs, intermediates, and drug products. So I just would like to give an insights what are the requirements of these high pure solvents in the manufacturing of drug products and intermediates and bulk drugs. Just I would like to give the quality requirements with respect to the regulatory authorities and with respect to the different grades of the solvents that are being used in the pharmaceutical world. So in Aragen, we have a purpose that in every molecule is the possibility for better health. This is our purpose, to work with every molecule, and we work with all the customers across globe, all the innovator customers and big pharma and emerging pharma customers. So this is my outline of the presentation. What is the role of organic solvents in our pharmaceutical industry? And I would like to cover all the team here who are participating right from the manufacturers of the solvents and for all the industries. But my focus, I would like to mention why the pharmaceutical solvents are more required on the purity requirements and how it impacts to the human health. So this will give a wider view how the solvent manufacturer or suppliers are contributing to the society and for their better health as well. So the quality of and purity of the solvents much more important, and I would like to cover that point also in brief. And solvent consumption in details, how much solvents are being consumed across the globe. Just I have given a ballpark figure. And the impurities present in the solvents, that is most important, wherein we track all these impurities which pose to the issues in the manufacturing of the bulk drugs and then pharmaceutical compounds, and the reaction byproducts and control of residual solvents and quality of solvent which is impacting the analysis itself also I would like to cover. So these are the different processes that we use the solvents, that is reaction media, crystallization process, purification of synthetic products and equipment cleaning purposes, and mobile phase in the Hplc analysis and chromatographic purification. These are the different activities that we do with wherein the solvents are involved. The organic solvents, which are the pharmaceutical industry is used to reaction media, yes, and purification and cleaning of the equipment, as I mentioned. And the solvent quality will impact even crystallization of the materials. That is most important. And the residual solvents present in the final drug product or drug substance would be tracked and then would be controlled, or it should be removed or reduced to the level. After drying of the material, we will check that particular pharmaceutical compound and then check how much quantity is present in that particular compound. And as per the guidelines, there are different guidelines, and then I'll just brief up what are the available guidelines to control those solvents, because the toxicity level of every compound, every solvent. And these organic solvents are also used in the analysis itself, in the HPLC analysis and all those stuff. But however, how pure those solvents are required and how it impacts our analysis also I would like to explain here. So this is a typical process, synthetic process, wherein different stages I mentioned for the API or even drug product. And these are the number of solvents usage which is showing here. The huge bulk solvents are being used very extensively in each and every step and then process or even isolation. So the quality or purity of the solvent is prime factor. A very large volumes of these solvents are being used in the manufacturing process, and it is essential that the impurities of the solvents are to be also minimized or to be controlled, if possible should be eliminated. So we are not only talking about the purity of solvents, but also the impurities present in the solvents are also most important. Why? Because these impurities will pop up in our drug product or the tablets which we are consuming day by day. And the solvents manufactured on the commercial scale must be the lowest possible impurities or below the specification limits. And I would like to give some insights on the impurity levels, how the regulators will ask us to control in the drug substance or drug product. And this would avoid the corresponding issues in the regulatory environment also, if we reduce these impurities, and then we can avoid so many issues with respect to the manufacturing process, as well as to improve the quality of the drugs that we are producing. And pharmaceutical companies require supply chain partners who can provide uninterrupted supply, but also with the consistent and reliable quality. This is most important because these solvents we are using very huge quantities. And if you see, this is the consumption of these solvents across the globe. The alcohols takes the major part, that is 34% out of all other solvents like amines, esters. Esters and ethers will have a same consumption levels, and the chlorinated solvents would have the less consumption levels. But also these chlorinated solvents have their own impact on the environment, as my previous speaker has mentioned, but also on the impurities levels. So impurities present in the solvents, as I mentioned previously, so how it impacts. Suppose if I take this methanol, and then this is a solvent, and if it reacts with the methyl chloride, this is a reagent which is being used in the process, and then which will eventually forms the corresponding impurities in the life cycle. You see different impurities. And again, if I use the ethanol, also again it reacts with the reagent and it will form some other impurities. So we should be very careful in our manufacturing process how the quantity of ethanol present in our process and how it impacts in the reagents also. So same way like ethyl acetate, if I take, and then it will produce the ethanol or ethyl acetoacetate, another impurities are also present here, and then that will also we will have to control. I have given some of the examples like THF, and then again if it is oxidized even at the room temperature or environmental conditions, this will form an acid. This is another impurity, so we should be careful in this also, and we track these. And again in the benzene, so again the toluene in benzene, so sorry, benzene in toluene, that is also most important, wherein the benzene is high toxic in nature. So I would like to take you some more insights in the next coming slides. So, and not only the impurities present in the as a solvents in it, and but also the reaction byproducts, like suppose if I take these tertiary alcohol, and then if these reacts with some of the reagents, this will form the genotoxic impurities. These are the different methyl esters, tosyl esters, and these are all the very genotoxic in nature and which should not contain in our drug substance and drug product. That means the solvents that we are using also would react and form a byproducts in our drug substance and drug product, and we should have to be tracking that. And if you see, suppose if I wanted to control this impurity, this is a genotoxic impurity, and I should have to control at the ppm level, let us say 0.3 ppm, 0.03 ppm level, it should be controlled in our drug substance and drug product. That means the impact of these solvents present in or the impurities or the byproducts of these solvents in our drug substance will have a larger impact for the efficacy and as well as the health of the patients. So how do we control these solvents in the drug product or drug substance, and how do we control these impurities? I would like to give, these are the authorities to protect the human health, and these are the two authorities, that environmental health criteria and integrated risk information system, IRIS, and these authorities will define the risk assessment of these residual solvents or the solvents which are being used in our pharmaceutical industry. And these are the regulatory bodies. If I take, these people will regulate all these all these levels of these solvents present in our drug substance and products. And these are the three different regulatory bodies in addition to the US FDA and United UPA and US EPA and then IPCS, that is Environmental Protection Agency, which is most important for these drug substance and drug product or the solvents intermediates and their quality. So how these exposure limits are established, that is as per the total daily intake of the solvent or the acceptable daily intake are being established, keeping in view of the patients use the pharmaceuticals and to treat their diseases. That is one of the parameters that these authorities would take. And the assumptions of lifetime patient exposure is also not necessary, but at least to check the daily exposure limit would be taken. But keeping in the consideration, yes, definitely the residual solvents present in the drug substance and drug product are unavoidable. Definitely they would be present in our compounds, but however their levels should be controlled according to the requirements or according to their exposure limits and the daily intake values. How it is to be determined? So there are classified as three different classifications, like solvents to be must be avoided. Definitely these are all some of the class 1 solvents must be avoided because of their toxicity and carcinogenicity as mentioned by my previous speaker. So, and as per the environmental hazards, and I would like to give what are those also in the next coming slides. And solvents to be limited to the required levels, and solvents with low toxic potential, which can be even with low toxic potential can have no health based exposure limits and can be used. These are all the three different classifications that authorities provided to us, and then we control according to that. So these are the different 4 solvents which must be avoided and then cannot be used. If these are all used, these 5 solvents, these must be controlled at these levels like 2 ppm, 4 ppm and 5 ppm. And this is most important for us, and we ensure that each and every, not only the drug substance and drug product and the solvent that we procure or purchase from the supplier must contain these class solvents must be less than the defined limits. And that's why we have a stringent controls in our solvent specification also with respect to these class 1 solvents. And then I'll show you which are the solvents contains these type of different reagents, or again these solvents present in it in my next upcoming slides. These are other solvents which has the defined exposure limits, and then these are the different limits. I don't want to go through each and every this thing, and then this is available in public. And these are some more other solvents which does not have any limits, but this can be used at the limit of 5000 ppm. And this is one of the typical method that we developed for a 16 solvents at a time, and then we can analyze within 20 minutes runtime. And then this is a typical chromatogram that I would like to depict here. And then these are the number of solvents which are commonly used in our pharmaceutical products and drug substances, so that we can control and track these solvents and their purities and according to their impurities also. And then I would like to show you one of the typical example during the analysis, how it impact, as I mentioned. So this is the baseline. This is the impurity present in the solvent, which is showing in a blue baseline. And then this is the impurity which is posing a challenge to our required compound. This is a required pharmaceutical drug substance, and wherein the impurity present in the solvent is giving a small peak which is showing and interfering in our analysis. And then if we avoid this, only then we can find out the exact how much pure it is. Otherwise, then it will be a challenge to us also to analyze the pharmaceutical compounds. So that's why again we have got another solvent which is that is the Hplc solvent. Now you see there is a clean baseline. That means there is no impurity in this solvent, wherein we can measure the exact purity of the desired drug substance or drug product, and then we can define the quality of the compound which is going to be consumed by humans. So this is most important. Even our drug analysis also, the quality of the solvent plays a major role, like even at a ppm level of any other impurity present in the solvent would cause the impact on our analysis. So I would like to summarize some of the points and the role of solvent in pharmaceutical life cycle of the pharmaceutical compound production, manufacturing and drug product. It is very important, and there are different grades of solvents like commercial grade, reagent grade, ACS grade. ACS means American Chemical Society, and then analytical reagent grade for the, as I mentioned, for the analysis purpose, and the commercial grade that we are getting in the bulk. But however, so the people will say this commercial grade is nothing but the pharmaceutical grade from the suppliers. We can say that. And then what is important in that? Not only the purity of the solvent that we are getting it, but the trace level of impurities would give a major challenge in our drug substance and drug product, as I mentioned in my previous slides. And what are those impurities? As I mentioned, the benzene, that is one of the impurity present in methanol and toluene distillation process, and it must be controlled to the 2 ppm level because of the toxicity of the benzene, as even restricted or directed by all the regulatory bodies across the globe. And in the EP monograph as suggested for the acetone, if you see benzene, methanol and IPA, these are to be controlled at these levels. That means we will be careful about our analysis also while receiving the sample itself, while receiving the solvent itself. We will check for these impurities present in the acetone. Acetone itself is a solvent, and the benzene and methanol are also solvents, but these are all impurities in acetone which carried forward during the distillation process. But these are all toxic in nature. Again, in the EP monograph of ethanol, if you take, there are 24 impurities presented in the monograph of ethanol, and it should be controlled to 300 ppm, because ethanol being extensively used in our pharmaceutical process, not only that along with the 24 impurities, and there is a specific impurity that was mentioned, because methanol is one of the carried forward impurity that must be controlled to the 200 ppm level, and acetol and acetaldehyde also should be controlled at 10 ppm level, because these are all the possible impurities that is carrying forward from ethanol. And benzene also, of course, should be controlled at 10 ppm level at ethanol solvent itself. But whereas it comes to the drug substance and drug product, it will be at 2 ppm level. And again, like that in dichloromethane, that is a chlorinated solvent that we use in our pharmaceutical process, that the carbon tetrachloride, again, also it is a class 1 solvent, as I mentioned earlier, and it must be controlled at the 4 ppm level. And some of the reaction byproducts, as I mentioned, due to the solvents that are reacting with our reagents present in the manufacturing process, also will generate a genotoxic impurities and subsequent corresponding impurities, which pose challenge the quality of the drug substance and drug product in the market. And that's why we are very much keen on analyzing the solvents and tracking the impurities of the solvents, and not only the purity but also the impurities of the solvents. Thank you. I'll take the opportunity to thank even the organizers giving me this opportunity. Thank you very much. And I'll take up the questions at the question answer section. Thank you very much.
Okay, thank you, Dr. Rao. That was very insightful discussion we had, and so the talk. I just like to point out few things that I also noticed, that you were mentioning high purity solvents while manufacturing of pharma products, and you mentioned the role of organic solvents. So it is very important for us to understand the impurities that are present in the solvents, and how reaction byproducts of solvents work. So that was one very important point you brought out. And at the end, if impurities are present in the solvent, so the main aim that we want to achieve is improving efficiency, like efficacy and health of people. So maintaining the impurities, say, or reducing them, or say reducing impurities in solvents, is going to help us enhance the overall health of the population, and that is the main aim. So two things that came to my notice was, we already have agencies like the US EPA and the US FDA, which is a good starting point for us to understand the regulations and the compliance and the standards requirements. It becomes easy for us to follow them, and they say bring it to, say, our country context or a different developing country context. That's a very good thing that we have understood. And definitely stringent controls is what are required to, say, improve the functioning of the solvents and coming up with better medicines in the future, for sure. Thank you so much, sir. It was very interesting to listen to.